Hey, what's up, everybody? Apologies again if you're watching this today on Friday. Uh, I'm sure most of you don't mind that we did a recorded lecture today and that you got to stay home slash whatever it is. I just know there was probably a good number of people who got up, got ready. You read that email while you're on the bus or sitting in the lecture hall. And to you guys in particular, I apologize. Yeah, I was planning on, despite the injury, still making it to class and doing it, but just some issues waking up this morning, realizing that yeah, I was going to put myself through a lot trying to get through that lecture. So here we are. We're going to finish up nervous system. Don't forget you got your lab midterm. Really hope you guys have been going to those open labs and doing some of those practice midterms. As Joe mentioned, that practice midterm thing is going to stay open during the week of the lab midterm. So if you guys can pop in and do some practice runs throughout the week, you should do that. Wiley number two is open, all that good stuff. Hey, really quick, just reviewing what we covered on the nervous system. First of all, the very, I would say there's really kind of two big things for this chapter on the nervous system. Number one, make sure you can organize the nervous system. So the table that I've given you, CNS, PNS, under PNS, sensory and motor, under the motor, somatic, autonomic, under the autonomic, sympathetic, parasympathetic. Make sure you can really just do that on a scratch piece of paper. Don't come to the exam without at least being able, to be able to construct that whole organization. Then, part two of this lecture is figuring out all of these cells, right? All of these neuroglial cells. So, just as a reminder, and I'm going to do some really terrible drawing today for you guys, satellite cells are the same thing as astrocytes, right? Astrocytes and satellite cells are both the exact same thing. Here they are right here. These are supporting cells that wrap around a neuron cell body, singular, right? One singular neuron cell body. So they're called satellite cells if that neuron is in the PNS. They're called astrocytes if that neuron is in the CNS. Same thing goes for Schwann cells and oligodendrocytes, right? Schwann cells are the same thing as, can I just put O? I can just put O, right? O, oligodendrocytes, right? That's the O one. Go back just a couple of slides, whatever. And here they are, right? Here's a Schwann cell right here. These are, they're similar to satellite cells and astrocytes. They wrap around a neuron. They get a different name because of what part of the neuron they're wrapped around, right? So a cell body, that's satellite cells and astrocytes. Here, wrapping around the axon, this little bubble wrap that goes around the axon, that's a Schwann cell or an oligodendrocyte. If that axon is in the PNS, it's a Schwann cell. If that axon is in the CNS, it's an oligodendrocyte, right? Same thing goes here, as we talked about. You just really take, I get the picture here. I'm just giving you one singular neuron, but we don't really do that very often. We don't take one neuron and go, okay, go out and do this really important job. Instead, we take this neuron, this neuron, this neuron. We just stack up a whole bunch of them and then bundle up many, many, many cell bodies. And if we bundle up a whole bunch of cell bodies, that's the part with the nucleus up here at the top, right? We take just not this one neuron cell body, but we take a whole bunch of neuron cell bodies, bundle them up together. If they're in the CNS, brain and spinal cord, right, they're nuclei. If we bundle up a whole bunch of those neuron cell bodies out here in, the rest of the body, those are called ganglia. By the way, those two terms are going to be really important, especially the ganglia, because next semester in physiology, you're going to have to learn about pre and post synaptic ganglia. Post ganglionic, pre-ganglionic, and there is a difference there. You're going to need to know, like, what are the neurons doing before they get to a ganglia versus after, and you'll get to do a lot more with ganglia next semester. But nuclei, ganglia, same thing here. If instead of taking that neuron cell body, we take this axon and we bundle up not just one axon, but a whole bunch of axons, right? But when we stack them all up together, we bundle them up. They're called tracks. If they're in the brain and spinal cord, they're called nerves if they're out here in the body. So when you do hit that funny bone, whatever, someone pinches you or whatever, it's not one neuron that's getting stimulated. You're stimulating Many, many, many axons all together. That's what a nerve is, right? Many, many, many axons running through your peripheral nervous system. So that's where we've been so far. We left off right here. I shouldn't even try, but I'm gonna do it because I think there's quite a few of you guys who are like, I would love to have this, build it out myself, come back to it. Now this is a recorded lecture. You can come back to it, right? I'm gonna take that in one color. And I'm going to take it in another color over here and put these are neuroglial cells that I'm drawing. These are cells that are wrapping around a neuron cell body. So if those cells are wrapped around a neuron cell body, if that neuron right there is in your PNS, those are called, hopefully you guys are good on this. I'm going to abbreviate all of this here. Satellite, right? Satellite cells. Hopefully you guys were good on that. And I know right now some of you are like, dude, this is going to take you all day. Can you not do this? And I get it. Like I'm committed now. I'm gonna shorthand these, right? Astrocytes. Those are astrocytes. Now, these cells right here, wrapping around the axon, a singular axon, those neuroglial cells right there are called oligodendrocytes, right? If that axon or that neuron exists in the CNS, correct? But if this neuron happens to live somewhere in your PNS, those individual cells around the axon are just called Schwann cells. SC slash Schwann cells. Schwann cells, right? Same thing. Oligodendrocytes and Schwann cells are the exact same thing. It's just where the neuron lives. Neurons in the CNS, oligodendrocyte. You're tired. You're tired of me. Well, you were tired of me before that. Let's be honest. Here we go. If we bundle up not one cell body, many, many, many cell bodies, many neuron cell bodies, those are going to be called in the PNS ganglia, right? That's a G. Yeah, that's a G. But if those big bundle of neuron cell bodies exist in the CNS, those are going to be called nuclei. Just go back if you want the rest of that, right? If we bundle up not one axon, many, many, many axons in the CNS, those are going to be referred to as tracks. Tracks. But I'm not going to change the color over here. You get the idea. You're going to, though. Those are called nerves. If it's a big bundle of axons out in your PNS somewhere, right? Not in your brain and spinal cord, out in the body somewhere, those are referred to as nerves. So please bring this table a better version with things spelled out and organized better, right? But bring this table to your next exam. Like I'm shocked at the number of people who don't take the time to make this table. You can imagine, right? Neuroglial cells surrounding the axon of a PNS. Big bundle of cell bodies somewhere within your cerebrum, right? Of the brain, right? You just boom, 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 check these things off really, really quick. You're going to get multiple questions on this exam, on the final exam next semester in physiology. And as mentioned, we're not done. We're going to get to the spinal cord today. I'm going to introduce it to you. I'm not going to try to draw the spinal cord and label all the parts, right? But I'm going to show you here in just a minute where we're going to get to and what we're going to do on Monday, how we're going to add the spinal cord to this table right down at the very bottom, right? So let me get back to where we left off. And we had a really, really good question when we were talking about the axon and we were talking about oligodendrocytes and Schwann cells. That's these guys, by the way. There's one right there. There's one of those cells. By the way, if I was to ask you right now, where does this neuron right here live? Where would you guess this neuron lives? And some of you guys are like, I don't even know what the hell you're talking about. CNS or PNS, right? Is this a neuron in your brain or is this a neuron in your foot? And the answer is on here. And some of you guys have already found it, right? It's right there. That's a Schwann cell. All these little cells that I'm showing you here, right? So this neuron has to live somewhere in your PNS. If those were called oligodendrocytes, it'd be a CNS one. So we got this question in class, being like, oh yeah, well, those cells wrapping around. There's something about plastic material, myelin, whatever, something like that. It has to do with MS. what's going on there. And that's exactly what we're going to talk about. That axon, this is a slide you've already seen before, right? That axon, that long tail-like structure is sending and receiving, right, back and forth. It's carrying electrical messages back and forth, back and forth, back and forth, right? And that's the point here. That's what we're going to get at. We're going to talk about these Schwann cells and oligodendrocytes and that material. And we're going to talk about what that material is. That material is called myelin, or what's known as myelin sheath. And that's something I think many of you have heard of before. If you didn't know, it is a mixture of both protein and lipids. We're going to actually explain why that is as well. And this is what gives us these terms, white matter and gray matter. Now, if you did not know, some of your nerves and your tracts, some of your individual neurons, right, in the CNS versus PNS, some of them are referred to as white matter and some of them are referred to as gray matter. And all it is here is that if a nerve or a tract or an individual neuron, in both cases, right, if it has this white material, this protein lipid substance called myelin sheath on it, it's going to be referred to as white matter. And when you do the brain dissection, as you're going to do in lab in like 2 weeks from now, I don't know, whenever you come back and do this after spring break, you're going to come in, you're going to cut into this brain, you're going to look, and hopefully your lab instructors will be like, hey, check it out. Find the white areas, find the gray areas. What's the difference between these two? And now you know, if it shows up as white, and it does on the brain, those areas have a lot of myelin sheath. Those are neurons that have what? Neuroglial cells, oligodendrocytes, right? If you look at the brain on that sheep brain and you look in there and there's like a glowing white area there, it's really bright. And you look at it, you'd be like, oh, check it out. There's a ton of oligodendrocytes, lots and lots of oligodendrocytes. How do we know that? Because that's here they are, right? If I can find my pen, there's there's one of these. Now, I get that's a Schwann cell because that's going to be out in the neuron out in the body somewhere. But let's just say this neuron is inside that sheep brain. That's going to be called an oligodendrocyte. It's just white material that wraps around that axon. And there's billions of them, by the way. It's not just like one oligodendrocyte. There's tons and tons and tons of these. Then on that same brain, right next to it, there's going to be an area that shows up. It's gray. It might be brown on yours, but it's just decayed, whatever material. But it's going to be a gray area. That area lacks myelin sheath. There's no oligodendrocytes on those neurons in that area. That's it. That's the difference. White matter, gray matter. Let's talk about it. Let's talk about why that is. Here's the reason. Here's the function of that myelin sheath. Why do we have it in some areas? And honestly, a more important question is why do we not have it in other areas? But let's talk about the why. Why do we have it in some of these places? Well, this is where if we were in class, this would go better. Sorry again. But I would go over to the wall where there's a light switch. I'd be like, okay, here's a light switch. Check it out. This is sort of how your neurons work, right? They get a message and then I push a button. And then you can imagine that electrical message, as soon as I push the button, it doesn't just stay on the light switch. Obviously, when you push the light switch, it turns on or turns off an electrical current that goes to the lights in the room. And so I push a button and the room goes dark, right? And back. And then the neurons get a new message, boom, and I push the button and the lights come on again. And that's exactly how it works. Your neurons connect to things like muscles. And when you want to move a muscle, you have a neuron that's like a wire and it goes from your control center. So this is where the analogy is going here. Your brain is like the button. And when your brain gets pushed and it says, oh, we need this muscle to move, you push a button. And then behind the wall, right, where the switch is, there's an electrical wire running to the light. Just like in your system, from your brain, you have an electrical wire running to every single muscle fiber. And when you say, okay, contract muscle, that's like pushing the button, electrical wire carries that information and it moves the button. So it's similar in terms of this setup. Now, if we're in the class, we're talking about this, be like, okay, but hold on really quick. What's behind the wall? And most people are, it's a wire, it's an electrical wire. I'm like, yeah. What's it made up of? And most folks know it's like some type of metal. Metal carries that current. But what wraps around it? And that's where I think a lot of people know who've looked at wires before have had to strip wires and wire things up. You have to strip off this plastic material in order to get to the metal. And then you can connect the metal behind the screw that you're tightening down or to another wire piece or whatever you're trying to do there. But you strip away this plastic. What is that plastic about? Plastic doesn't carry the electricity, the metal does. So why do we take almost every single electrical wire and wrap it in plastic? And those of you who know about electricity and worked with electricians or whatever, you know the answer is right here. It protects and insulates the electrical message. And that's exactly what myelin sheath is. It's just like plastic wrap around wires in the wall, connecting your light switch to your lights. Now, here's the question. How many of you have accidentally stripped away some of that plastic material or touched a wire that was on and you touch it on the plastic part, you're okay. But how many of you guys have had wires that were exposed a little bit and you accidentally touched it when it was on? You got some of that electricity. You got some of that electrical message. It's running constantly back and forth between, I don't know, the light and the light switch, source and the sink, whatever it is here. When you touch it or get close to it and make contact, you grab some of that electricity and it goes to you and it hurts. It hurts, by the way, if you've never done that before. Don't do it. It's your one lesson today. Don't do it. But all right, so same thing exists with neurons. Why do we put electrical wrap around wires? It insulates. It keeps the electrical message intact and running smoothly. All that good kind of stuff, right? And what happens when we peel off some of that electrical or that plastic wrap around the electrical message? We can lose. Some of you, many of you guys have been somewhere where there is an issue and the electricity isn't running properly. The lights, they flicker when you turn the light switch on. It's like it takes a second to come on. It's inefficient. So let's go back to these neurons. Why do we have the myelin sheath on there? To insulate and keep the electrical message running smoothly, to keep it from breaking down. What would happen if a neuron was supposed to have myelin sheath, but didn't, or didn't have the right myelin sheath, or degraded myelin sheath? That was the question that we got in lecture the other day. And I'm going to show you a video here. Hopefully it shows for you properly. Just this is sort of like a trigger warning for anybody. There are people almost always who have a family member or know someone with MS and it really hits close to home. I'm going to show you a video of someone with MS and show you what it looks like. And so if you don't want to watch it, you can kind of skip ahead or whatever if it's just kind of hits a little too close to home here. If you didn't know, MS is a debilitating genetic disorder. We're going to talk about it a little bit more. But when you are diagnosed with it, they give you lots of these help videos and they show you different people at different stages of MS. And this is someone who's been suffering with MS for a little while. And what they're helping someone else who's been diagnosed, who's trying to figure out what to expect. Here's what it looks like getting dressed when you have this stage of MS. Now, what you'll notice is muscles are working in some parts of his body, but in the muscles that are further away from the brain, the muscles that have neurons that are traveling longer distances, those muscles are being lost. And that is MS. That's what's going to happen. You are going to lose more and more and more of your muscle function. That's what's so terrible about this disease, right? There's no real cure for it. And it's a slow degrading of this. They're going to lose more and more and more of these muscles. So I'm going to move from the video here and take you to the answer to this. So what's the difference between that guy in that video and you? Here are your neurons traveling to muscles, and there's your myelin sheath. The guy in the video, that's his myelin sheath. MS is a genetic disorder disease in which the protein and lipid material that makes myelin gets degraded. That's it. That's it. There's no other. They just have this genetic wiring that is going to attack and slowly degrade the lipid and protein substance around all of those neurons. They don't lose all of their muscles, boom, at once. It's not like paralysis. It's a slow degrading and losing of this message. Why is it that the legs are going to be one of the first muscle groups to go and to be lost? This is the longest distance from the brain. And they have far more myelin sheath being lost. So let's go back to if you were ever younger, whatever, slash the other day, and you saw a wire where some of the wire was exposed, and you're like, oh, I wonder what would happen if I touch it. That's sort of what you're simulating here. The plastic wrap is being lost and other tissues are sort of touching this. And so some of that electricity is being lost. You're losing the strength of that message. And eventually all of that myelin sheath is going to be gone, maybe not all of it, but enough of it that enough of the electrical message is being lost in other tissues or isn't being transmitted to the right locations. And you are eventually going to lose the use of those muscles. That's MS. Really debilitating, terrible, terrible disease of which there's no cure for still. Those of you who are considering going into that area and doing research on this, kudos to you. Definitely need more research on that. So that's nervous system. Again, two major parts here. Organize the nervous system, that table, and then make sure and organize your CNS, PNS, astrocytes, satellite cells, oligodendrocytes, Schwann cells, right? Those are the two big parts. This is just a friendly reminder. Do not forget, you need to know these A and E directional terms. Sensory is always afferent because it's going towards something. The towards is your brain. And efferent is always out, right? The opposite direction, leaving and going back out towards your effectors like muscles and glands, all that good kind of stuff. Make sure you know those things. Okay, so all I'm going to do here with the rest of this is sort of give you this little teaser to the spinal cord. It's where we're going next, right? Going to the spinal cord. I can't. I can't do things at the same time. So here we go. Let's just touch on spinal cord. We're going to cover the spinal cord pretty quick on Monday. So I just want to introduce you to what we're going to talk about here. Spinal cord is more than just a continuation of the brain. I know that's what most people think. Like there's our brain sitting up top, and then we just have this long thing that's basically kind of like the brain, I think, that's just a highway, right? And you're not wrong there. It does connect directly to the brain, and it is a highway. We're going to relay those things. So when you do hit your funny bone, when you do touch something really, really hot, it doesn't go directly to the brain. Instead, it travels up and down through your spinal cord. Here's what's really great about it. We are going to take that message of really, really hot. We should move our hand, and we are going to speed this thing up and get it to exactly where it needs to go. That's what your spinal cord is going to do. We're not going to rely on even a big bundle of mini axons. No, 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 no. We're going to put this thing into a tract. We're going to put it into a separate organ called the spinal cord. And you know what we're really going to do? This is what's so amazing about it. When you touch something hot over here, and then immediately decide to move the muscle over here, right? Those are two different messages on the spinal cord. We're going to take sensory, ow, I touched something really, really hot, and we're going to put it on one side of your spinal cord, actually back here. And then when your brain goes, oh, move these muscles over here in the same arm, right, that was just picking up on this message, it travels on a completely different, we separate out sensory over here, motor over here. That's what your spinal cord does. It keeps those two messages completely separate. Sensory, motor, and as a result, it speeds up, gets the message there faster, and comes back. How do we separate those two out? We'll talk about that. When we go into the brain and the spinal cord, we have to talk about, can't find my pen. Don't think I'm going to have a pen on this one. Maybe. What's this going to do? Yeah, I got a pen. It's a different pen. I don't know where that one came from. We're going to talk about meninges. Meninges are covering. I was gonna write covering, right? Or wrapping. They're coverings and wrappings that are CNS. I will write that part. Meninges are CNS structures. These are wrappings, coverings, Some of you are like, dude, sometimes your Arkansas really comes out. Wrappings, we're gonna put wrapping around around our CNS structures. Your entire spinal cord and your brain does have these protective coverings, similar to like the epimysium, paramysium, endomysium. We're gonna do the same thing here. Meninges, three of them as well, are going to be three different coverings that go around the brain and the spinal cord. Now, these prefixes will help you out. Dura, right? For dura, your dura mater. First of all, that prefix dura is the same thing we use like endurable. Its ability is very enduring, right? Is sort of how that translates to. But dura means durable. That's what I want you to think of there. This is the outermost covering, right? Outermost covering. We can see right here. I don't know really how to change colors on this one. I don't know what's going on. Why I got a brand new thing here. Let's go with red on this one, arachnoid. And your arachnoid layer is going to be this next layer here, this more avascularized layer that we're going to see right there. And then eventually your pia mater here. The pia mater is the delicate. That's what the term means. And it's going to be this real thin layer inside here that's kind of being peeled away. And we're going to see these again on the brain. Just want to introduce them to you. Three protective layers. The matter literally translates to mother. It's trying to, they wrap around like a mother protecting a newborn, whatever. That's sort of the idea here. Matter means mother, a protective mother covering here. And then each one of these prefixes means something else. Dura is very durable. It's on the outside. It's really, really tough. Pia, the last one there, the innermost layer, means delicate. Delicate, right? And then arachnoid, it means what you think. It's spider mother. Like I don't know. But if you take a fresh brain or spinal cord and you peel off the dura matter, you do get this real cobweb-like material. It's weird. You might be able to see it. Ask Joe to pull out some of the sort of fresh brains that have been preserved. And maybe if you peel away at different parts, you'll be able to see this. But it does look like cobweb type material. We'll do it again with the brain. But no, there's three protective layers: dura matter, arachnoid matter, pia matter. Inside of that, I don't know how. I don't know how anymore. I don't know how to get this to go. Can I do this? Will you go? Yep. There we go. Oh God. You can laugh. I don't care. I've never used this one before. I've never gotten this far before. Uh, sorry for that. I think you should leave, fans. You got that one. Uh, so great thing about this slide, you don't have to know it. You don't have to know it. You don't have to know any of this stuff on here. We're just going to move right off of it. I keep it in here to highlight. Like the spinal cord isn't just this one giant tract, right? I mean, it kind of is there, but there's many tracks going up and down. There's a lot of working parts. Your conus medullaris. Some of you guys, I'm not going to draw on this. I was just going to circle conus medullaris if you want that one. Some of you guys who work in certain clinics that deal with X-rays, MRIs, and things like that, you've probably seen that term for. This is the cone. This is where everything's sort of coming down in the spinal cord. It's near the end. The filum terminal is going to be this portion at finally the very end. It goes into the coccyx. It leaves the spinal cord and goes out to the rest of the body. There's a lot of ligaments. There's a lot of working parts to the spinal cord. Those of you guys who want to go into that field, uh, really cool stuff, right? This is all I'm trying to do. I'm trying to get here and get to the end of this. Let's draw one more time. This is the structure I'm trying to get to. The whole lecture over the spinal cord is going to be dealt with right here. A transverse cut through the spinal cord. We are going to deal with these structures inside. See, it looks like a butterfly, but it's gonna get this letter H, right? It looks like a letter H as well. That is your H. Those are the horns. The horns. And notice around just outside of them, really, really white, right? Really white. Lots and lots of what cell is in that white area, all that white area? Oligodendrocytes. There's a lot of myelin sheath made by oligodendrocytes. Why oligodendrocytes, not Schwann cells? We're in the spinal cord. CNS, right? We're inside the spinal cord, so these are CNS. But the H, that the big horns that we can see there, all of that's gray. No oligodendrocytes, right, in that area. So we're going to deal with those horns right there. And we're going to talk about, and what we're essentially going to do is deal with a posterior side and an anterior side. And we're going to differentiate those two. We're going to draw that picture of the spinal cord on our CNS PNS thing with the astrocytes, satellite cells, nuclei, ganglia. We're going to add this picture sort of the spinal cord down at the bottom, and we're going to add in horns are inside the spinal cord, so they're going to be on the CNS side. And then on this next slide, the coloring is going to be there. It's going to be there. Over here, these structures are the roots. The roots. There's a root. Here's a root. There's a root. I don't know why I'm saying it like that, but there it is. You have roots coming off the side. Those are your PNS structures. So why are you doing this? Sounds like you're about to finish. I am about to finish, but I'm giving it this year for the people who are like want to study over the weekend. I don't like if you want to get ahead and get in on this, like check this out. This is where we're going, right? Your horns and your roots. And they're on there for the next several slides, right? And just look ahead if you want to look ahead. There's your horns. Horns are inside your CNS structures, and we're going to separate the dorsal side from the ventral side, sensory side, motor side. Then we're going to go to your roots, and your roots are PNS structures out on the outside, sensory side, motor side, ventral, dorsal, right? We're going back to that. There you go. There's all your coloring in the right place. Okay, that's all I'm going to do for you today. That's all I've got. I may never get off this slide, though. This recording may go forever. There it is. Okay, I'm going to stop there. We'll pick right back up on the spinal cord, get through it really quick on Monday, and then we're going to go right into the brain. Oh, it's the best. The brain's the best. You don't want to miss it. I'll shut up. See ya.